faculties and non-teaching staff members of Electronics and Communication Engineering Department for the webinar. I would like to welcome our chairman, Sri S. Mariswamy, Sri A.R. Krishnamurthy, Secretary and Managing Trustee, Dr. B. N. Umesh, Treasurer, Sri P. L. Nanjundaswamy, Trustee, Sri S. Shivamalu, Trustee, and Dr. M. Mahadeva, Trustee of our PVP Welfare Trust in their absentia. On behalf of PVP Welfare Trust and Dr. Ambedkar Institute of Technology, I would like to welcome today's speaker, Dr. Humaira Nisar, Associate Professor, Head of Program, Master of Engineering, Electronic System, Faculty of Engineering and Green Technology, University of Ranko Abdul Rahman, Malaysia. I welcome you, madam, for the webinar. The gist of webinar is like this, what I know I would like to be sharing. Neurofeedback is also called as electronic encephalography, that in medical we call it as EEG. A non-invasive way to control and measure your brain waves and body temperature. This form a therapy allows your neurofeedback, which provides your brain waves through conditioning. Neurofeedback training means it teaches self-control of brain activity to the subject by measuring brain waves and providing a feedback signal in a real time. These findings provides a strong rationale for use of neurofeedback training in the resolution of response conflict phenomena in health of human being. So what is this neurofeedback therapy used? It is also called, as earlier I told, EEG. That is, biofeedback or neurotherapy is an adjective of treatment used for psychiatric conditions such as attention deficit, hyperactive disorder, generalized anxiety disorder, post-traumatic stress disorder, phobia disorder, obsessive compulsive disorder, bipolar disorder. What are the feedbacks of this neurofeedback? Patients report enhanced memory and focuses, decreased impulsivity and anxiety, better mental clarity, more restful sleep, improved mood, and host of other benefits after using neurofeedback without side effects. So can we ask a question here? Can neurofeedback be harmful? As a general rule, most of neurofeedback clients experience no adverse effect from neurofeedback when done properly. Generally, most people have no adverse reaction to neurofeedback sessions. So there is one more thing, that is brain mapping can be used for. So that the brain mapping is an important tool we use to evaluate your brain waves and identify opportunities to improve communication between various regions of the brain. So neurofeedback results permanent. Once it is properly done, the new patterns, if the brain is accepted, it results will be last. So with this small information about the neurofeedback, now I would like to thank Dr. Ramesh and the coordinator of webinar, Ms. Shweta N, Dr. Shilpa K.C., Mrs. Sangeeta N, and Mr. Siddesh for arranging the webinar, Effect of Neurofeedback Training on Response Conflict. Nowadays, if you see, the people are stressed due to present condition. This webinar may help to understand brainwaves and response reasons to overcome stress and anxiety with the help of this EEG equipment, which they are going to discuss by the eminent speaker today. I also thank all the faculties and technical staff members of the Department of Electronics and Communication Engineering. Finally, I hope all the participants will come to know the effect of neurofeedback training on response conflict. Thank you, Ramesh. Over to Shilpa.
Thank you, sir. Thank you. I now request Sangeeta, Assistant Professor, Department of Electronics and Communication Engineering, to introduce the speaker of the day, Dr. Humaira Nisar.
Thank you, ma'am. A very good morning to all. Myself, Sangeeta N, Assistant Professor, Department of Electronics and Communication Engineering, Dr. Ambedkar Institute of Technology. I take the privilege of introducing the speaker of the day. Dr. Humaira Nisar has a BS Honours in Electrical Engineering from the University of Engineering and Technology, Lahore, Pakistan, MS in Nuclear Engineering from Quaid-e-Azam University, Islamabad, Pakistan, another MS in Mechatronics and PhD in Information and Mechatronics from Gwangju Institute of Science and Technology, Gwangju, South Korea. She has more than 20 years of research experience. Currently, she is working as an associate professor in the Department of Electronic Engineering, University Tunku Abdul Rahman, Malaysia. She has worked as the head of the Master of Engineering Science program for 5 years. Currently, she is the headmaster of Engineering Electronic Systems program. Her research interests include signal and image processing, image analysis for wastewater treatment, biomedical imaging, neurosignal processing and analysis, brain-computer interface, and neurofeedback. She is a senior IEEE member and she has published more than 150 international journals and conference papers. On behalf of Dr. Ambedkar Institute of Technology, I welcome you, madam. Now, I hand over the session to the speaker of the day, Dr. Humaira Nisar. Thank you.
Okay, thank you very much for the very nice introduction. And I would like to thank Professor Rajendra, Professor Ramesh, as well as all the other members of the faculty who have, I find it very privileged to have the invitation from your side. And in addition to this, I will really like to thank Ms. Savita, who had been coordinating with me for a very long time, and she has been very nice in her coordination. In addition, I would like to thank Ms. Shilpa and Sangeeta for arranging this very nice webinar. So, without further delay, I would like to start my talk. So, I think I need to share the slides first. Okay. Okay. So let me share. So can you see my slides?
Yes, ma'am. Yes, ma'am.
Okay. And
It's presenting.
yes, ma'am.
Okay, now?
Yeah, it's perfect, ma'am.
Okay, it's perfect. Let me turn off the video. Is it okay if I turn off the video?
Yeah, it is okay, ma'am. It's not visible actually now.
Okay. So, all right.
The screen is not visible, ma'am, actually.
Yeah. Just a minute. Okay, now it's visible.
Now it's fine, ma'am.
Okay, all right. So thank you very much for the very nice introduction. And let me start with the presentation. So the title of my talk is the effect of neurofeedback training on response conflict. So as introduced, I am Humaira Nisar from the Department of Electronic Engineering, Faculty of Engineering and Green Technology, University Tunku Abdul Rahman, Malaysia. Okay, so these are the contents of my talk. So I will start with a very brief introduction so that we know that what are the key terms in my talk, and then we will move to the methodology. So the methodology will cover the experiment design, data acquisition, followed by the data analysis. And then we will move to the results and discussion section where we will see the effect of neurofeedback on the cognitive ability, which will finally, I will conclude this talk. So let's start with the introduction. Okay. So what is electroencephalogram or EEG? So what is EEG? So EEG is basically the electrophysiological signal that records the brain activity in terms of electrical potential. So this signal is basically generated by the firing of the millions of neurons together. And very important to note that this is in the range of this electrical activity is of the order of microvolts, right? So let's look at this cross-sectional view of the human brain. You can see this is the brain inside, which is covered by our skull, right? So this is our skull. The top surface is our head, and the electrodes for EEG acquisition are basically placed on the scalp, right? And you will see that here you can see that the electromagnetic field generated by the neurons is present here. So this electromagnetic field is radiated back to the scalp, and where it is captured by the electrodes in the form of an electrical, you can say the electrical signal, right? So this is basically the EEG signal. So you can see that this EEG signal is not a very simple signal, because the skull is a 3D structure, whereas the scalp looks like a 2D surface, right? So the current which is generated, the activity which is generated inside the 3D structure is radiated on the 2D surface, which means that if you have some activity here, it can be radiated here and here as well. So that's why it makes a complex problem. We don't have a one-to-one relationship between the EEG generation and the EEG collection. We don't have a one-to-one relationship, right? The relationship is a bit complex. So anyway, so this is basically the EEG that we have, and it was first recorded by Hans Berger in 1929. So almost a century, right? EEG recordings. So when EEG recordings can be non-invasive. They can be invasive also, but here we are talking about the non-invasive EEG recordings. They have a very high temporal resolution depending on the sampling rate of the device. And nowadays we have some low-cost devices also which can be used to capture the EEG. Let's move to the next term, which is the neurofeedback training. So what is neurofeedback? It is basically a procedure in which the brain signals are utilized to train the participants to learn self-regulation. So your own brain signals will be utilized so that a participant can learn self-regulation. That is, the participant will learn that how he can train his or her brain activity. So how we do this, it is the setup is something like this. For neurofeedback, the participant is sitting in front of a you can say a monitor which provides a feedback to his brain activity. And how it provides the feedback, we will see this in detail later, but the EEG signal is being recorded and it is transferred to this after lots of signal processing. It is transferred or displayed on the screen. On the screen, the signal is displayed in real time. You can see this small bar of red color. So this bar will be fluctuating if the signal is increasing or decreasing. And the participant will be trained to either increase or decrease the signal depending on the protocol design. So this is basically known as the visual neurofeedback. So participant will know that his brain signal is increasing or decreasing, and whether he has to further increase it. So he will use different strategies to increase this signal. Okay, so this is basically the neurofeedback training. And so if we go back to the description, how we do it, we know that the brain has different frequency bands. So let's say a specific frequency band, for example, alpha band or alpha power of the signal is extracted and it is displayed on the screen. And the participant will try to increase or decrease this power, and in this way he or she will learn to control the brain activity. So this is what we call the neurofeedback training, and you can see that it looks like that it is very harmless. To date, we have not heard that there are any adverse effects of this training. Okay. So this is basically the screen for the neurofeedback. Okay, so now we will look at, there is another term in my title, which is response conflict. So what is response conflict? So if we talk about response conflict, we know that sometimes in our daily life also, we have to make choices that go against our learned habits or natural response tendencies, right? For example, very simple example is you want to move a lever to the left, but you have to make a right turn, right? So you have to move the lever left, but you have to take an opposite turn. So these situations generate response conflict. Okay, means you have to think differently. You have to take the opposite decision. So individuals with better attentional control will easily resolve this congruency effect. This response conflict may also be represented in terms of the congruency effect. So how we will be able to because everyone wants to, in our daily life, we have to settle many issues which causes conflict, right? So how we can handle this conflict in a good way is that if we have good attentional control, and how we can improve our attentional control. Better attentional control can be achieved by having high activity in the brain, in the frontal region of the brain. So you know that the brain is divided into 4 regions. So the frontal region is the region which is above the forehead. So if you have better alpha band activity in your frontal region, then you will have better attention, which means that if you have better attention, you will be better in the response conflict. Okay, so that's why we can use neurofeedback training to improve the activity in the frontal region. And when we will improve the activity in the frontal region, we will improve response conflict as a cause and effect example. So now we will look at the relationship between NFT and response conflict. Basically, NFT is you may have heard, it's very commonly used for the improvement of cognitive abilities. So what are the cognitive abilities? These are the basic things that we do in our daily life. Like we need attention to do any kind of task, right? We need memory. We need to improve our memory. We need to resolve the conflicts. So all these things come under cognition. So the improvement of cognitive abilities will aid in the processing of resolution of conflict, and NFT will help to improve the cognitive abilities like memory, attention, response conflict, problem solving. These all come under cognitive abilities, which we use not only. It's maybe sometimes people think that the people who are steady, or they only need the cognitive abilities. No, everyone needs the cognitive abilities. Like you wake up in the morning and then we follow a routine. So all of us here are not very old, right? So we have no problem in following the daily life activities, right? We know that we wake up, we get ready, we know that where is our car keys, and we can drive, go to work, right? But you may have heard that when people get old and they have this decline in cognition, then what happens? You forget where your car keys are, right? And you forget simple procedures, how to do simple things, and just like in the case of Alzheimer or these dementia, these diseases, what happens? The cognitive abilities start declining. So NFT can be used to not only for those people who have these neurological disorders, but with old age, the cognitive decline comes. So NFT can help to improve the cognitive abilities, not for only the healthy subjects, but also for the people who are getting old. So what we are going to do that in this work, we will use alpha protocol to improve cognition. So we will see that what is alpha protocol very briefly. So here we will see the different EEG frequency bands. So we know that the brain waves work in the range of around from 0 hertz to, you can say, 1000 hertz. And this frequency is divided into, you can say, 6 or basically 5 frequency bands. The mu frequency band has the same frequency as the alpha band, but it is present in specific position on the cortex, like the sensory motor cortex, it is present. So basically these are 5 EEG frequency bands. And when we talk about these frequency bands, you see that the delta band has a frequency range of 1 to 4 hertz. It is present in specific locations. It is associated with a specific task. When we talk about the alpha band is in the frequency range 7 to 13. Now there is another discrepancy here. To date, the scientists have been unable to resolve this matter of frequency ranges. If you will read lots of text, you will see that here you see that I have written alpha is 7 to 13, but in the rest of my my slides, you will see that alpha, the work that we have done, we have taken alpha from 8 to 13. So the scientists have yet not reached to a consensus that where one frequency band starts and other ends, because it's a very complicated, I think, task, right? So that's why sometimes it is also not possible to redo or reconstruct all the experiments or all the research that has been done, because different labs will be using different frequency ranges, right? So anyway, so now we will look at these frequency bands, and we will see that how these frequency bands are related to cognition. So it is believed that each of these frequency bands is associated with the cognitive functions. Like if you talk about the alpha band high activity of alpha band results in the relaxed state. And when you are in the relaxed state, they say that your cognition automatically improves, right? Because if you are stressed, your cognitive, you may experience some cognitive decline. Similarly, if you talk about the beta band is from 13 to 30 hertz, and it represents high level cognitive functions. When you are problem solving, or let's say you are doing some, you are studying, then you need concentration, and your beta activity is high at that point. So it is believed that each frequency band is associated with some specific cognitive functions, and after successful modulation of of the chosen band activity will lead to the enhancement of that specific cognitive function. So here in this study, we will be using the alpha protocol or the alpha frequency band to enhance the response conflict task. Okay, so let's move to after the introduction of the frequency bands, we move to the literature review. Lots of literature is available on neurofeedback. So this is the summary of the literature, and you will see that based on this general literature review, we see that the NFT may improve the alpha asymmetry, which results in a decrease in anxiety, right? Then you also see that gamma NFT results in higher test scores. Okay, here you see that again, alpha asymmetry could be improved by NFT protocol. Alpha asymmetry means it basically results in the improving of relaxation. So these are the general studies, and now we move to the summary of the studies using the beta protocol. The beta protocol is from 13 to 30 hertz, right? And you see that these are some of the publications. Okay, specifically when we use NFT, then sometimes we treat the specific region of the brain, right? So if we treat that specific region of the brain, increase the activity of the specific frequency there, then we can enhance the cognitive activity. So these are basically what they want to do in these publications. They want to enhance the low beta activity. The beta activity can be divided into low to high, means from 12 to 15 is low and 15 to 18 is high. And then these are the number of sessions, which in these studies, the number of sessions has been used. Now you will see that in every different study, the number of sessions is different. So again, that's why the research is not finished in neurofeedback. Still, it is at a very early stage, because there is no consensus that what kind of protocol protocol will improve which kind of cognition, to what extent, how many sessions will be used, what will be the number of sessions, what will be the duration of the sessions, right? And which site of treatment will be used. So different studies show different kinds of arrangement, and still that's why this is a very open research area. And if we look at this alpha protocol, we see that what is the outcome? Beta protocol, the outcome is that improvement in attention, right? Attention has improved, cognitive performance has improved, improvement in attention and intelligence. So these are very good outcome of this beta protocol. And now if we move and look to the alpha protocol, again, the organization of this slide is the same. Here we see that when we use the alpha protocol, which is basically from 8 to 13 hertz. Now, if you look at these publications, most of them use the alpha frequency range from 8 to 13, but this one has used 7 to 13, right? So, and then the number of sessions are also not consistent. It is as low as 1 session to 30 sessions, so very high number of sessions. Anyway, the outcome is somewhat similar in all cases. You see that it results when we use the alpha protocol, it results in the improvement of the cognitive processing speed and executive functions, improvement in cognitive performance, enhancement of cognitive performance. So it means that alpha protocol gives us good results. And then the site of treatment when we use the alpha protocol is normally we use the a site of treatment is the frontal region of the brain, right? So then we also see that the advantage of the alpha protocol is not only improvement of the cognitive performance, but it also helps to decrease the anxiety, right? Nowadays, everyone is very anxious. So this is another advantage of alpha protocol. So based on the literature review, we see that there is some positive effect of neurofeedback training using alpha protocol, and we want to enhance the cognitive function of the population, right? The general objective is so now I will move to the objective. There are many objectives of this research, but for this talk, I have specifically chosen this objective to explore the brain network and spectral changes associated with neurofeedback training for response. We will which helps to resolve the conflict based on better attention. And how we can achieve it? We can achieve it using the alpha-based neurofeedback training. So we will try to improve the alpha frequency range in the frontal region of the frontal region of the brain. Okay, and then we will induce some conflict. We will be using the Stroop EEG analysis. Stroop task is basically a psychological experiment which helps to introduce the conflict inside the human brain, right? So we will very soon we will see that what is the Stroop task. From the objectives, this is our objective, and now we will move to the methodology. So what is the method that should be adopted to carry out the neurofeedback training? So it is a bit not complex, but it is a long process. So if we look at the block diagram, we will see that we start with the study with the literature review, because we need to know that what we want to do and what previous people have already achieved, right? So after literature review, since this kind of a study needs the human participants, so we have to go for the ethics approval. For ethics approval, we have to design the experiment first, because in the ethics approval document is a very long document, and you have to show the experiment design as well as the number of participants you will be hiring for that specific experiment, and the details, all the details of the experiment and the participants. So after ethics, along with the experimental design, you have to get the ethics approval. I believe that every university has this ethics approval committee, which I think they work very efficiently, because our subjects are healthy subjects. If you decide to hire subjects which are non-healthy subjects who have some neurological disorders, like if you want to work with a hospital, which is a very exciting work for me also, but it takes a longer time to get ethics approval, because you have to obtain the ethics approval from the related ministry, right? For our case, we just our participants, we are healthy subjects which we hired from our own university, right? So we only got the ethics approval from the university committee. But if you want to work with some hospital and the real patients, then you have to get the ethics approval from the related ministry, right? So that's why you need to plan your research beforehand, right? Before you hire a research officer to work on it, you have to keep all this in mind, that this work takes lots of time, right? And once your ethics approval is you got the approval, then you start hiring the participants. But of course, you make all the plan beforehand. Once you hire the participants, you have to screen the participants, and that based on your studies design, like because we wanted the healthy participants, so we check that the participants don't have any underlying conditions, any disease or any neurological disorder, which is followed by of course they have to sign some consent form. So this is all the administrative work, and once we have finished this, then the neurofeedback training starts along with the data collection. So for our for this study, we collected the data for about 30 sessions per participant, and it took around 3 months to 2.5 months for data collection. And then data collection is followed by data organization, cleaning, and processing, because this is a very big amount of data. So you have to store it very efficiently, so that you don't forget what you are doing. And once the data is organized, then you have to clean and process it. Cleaning means removing of the noise and the artifacts. And then after that, the processing starts. Once you process the data, after you process the data, then you have to analyze the data, prepare the results, and of course, we have to write some reports, and we have to prepare some manuscripts. So this is basically the block diagram of the study, and now I move to this is the general block diagram, and now I move to the experimental design of this particular task. So I move to the next slide. So this is the experimental design for the NFT module, because we need to design the NFT module. In the market, you will find pre-designed NFT modules also, but they are very expensive modules, right? So it is also possible that you design your own module. So how we design our module, very briefly, I will tell you that. So based on this block diagram, you will see that. This is the participant who is sitting in front of the computer. He is wearing the headset, the headset to acquire the data, EEG data. So this headset is the EEG data goes to the researcher's PC through this is a Bluetooth based data, and then from here you see that. all the channels are being recorded over here. So it means that this is the raw data which is being stored on the computer. Now we don't need the raw data, because this data consists of, let's say, 0 to 60 hertz of EEG, right? But we only need the alpha band. So it means that we have to do lots of signal processing. So how we will do the signal processing is that we have this is the Open Vibe software, which is a free software. It is a BCI software, so it can do all the signal processing, which means that you have to decompose your data and filter it from the noise. And since we need the alpha band, so we will extract only the alpha band, which is from 8 to 13 hertz. So once we have extracted the alpha band, then what we are going to do, we are going to calculate the alpha power. Okay, so we will calculate the alpha power. Okay, and how we have to display this alpha power on the computer screen in the form of a bar. And this bar should be displayed in real time. So how we are going to do this? Because OpenVibe cannot display the bar on the computer. So we have to connect it with the Unity 3D design software. This Unity 3D design, those people who like to design games, this is basically a gaming software. So what we do is basically we extract the alpha band, and then through the Unity 3D design, we have just a very, very simple design. We have designed a red vertical bar. This red vertical bar will, the value of this bar will change. Okay, so how we are going to do this? The power values within the alpha band, as I mentioned, is 8 to 13 hertz, and these are taken not from all electrodes. This device consists of 14 electrodes, but we have chosen only the four frontal electrodes to compute the alpha power. Then we will be using a 5-second sliding window for power computation, which is updated every second. So this bar is updated every second, and this value is shown to the participants. So participants will know in real time that what is their alpha EEG activity, and they will try to improve this activity. So this is basically the design of the neurofeedback module. So once we have designed the neurofeedback module, then we need to know that how we are going to measure the response of the conflict, right? So we have been using the Stroop task to measure or to introduce conflict and then measure its response. So what is the Stroop task? The Stroop task is a psychological experiment which displays an stimulus on the screen. The stimulus is basically a word written in a color, right? So the participants must identify the color of the word. Okay, so what you have to do, this is a word written. This is known as the stimulus. This is the green, right? It is written, the word is green, and in this case, the color is also green, right? So this is first condition. The response is that the participant has to say the color of the word. So the color of the word is green. So the participant gives a very fast response, green, right? Okay, so this state is the congruent state in which the color and the word is same. So now let's look at the condition B. The word is green, but the color is red. The participant is supposed to say the color of the word. So he has to say red, but you know that according to the human visual system, or you can say the human brain cognition, we speak the word quickly rather than saying the color. So it may be possible that instead of saying red, you will say green, right? So that's why the response is slow. So this condition is known as the incongruent condition. Incongruent means the color and the word are different. The congruent means the color and word are same. So this difference in the color and the word will introduce a conflict in the brain, right? And that's why your response is slow. So if you have very good attention, very good cognitive skills, so you will be able to resolve this conflict quickly and accurately. Okay, so this is basically known as the response conflict task, and we call it the Stroop task. Okay, so now I will go into the details of the Stroop task. We have used the computer version of the Stroop task, and we call it the Stroop color word test. We have designed this task in the psychology experiment software, very common software. It is known as the E-Prime. It is very easy to design any kind of experiment, a neurological experiment using this E-Prime software. For the Stroop task, we have used 4 colors: green, red, yellow, and blue, and they were presented on the light gray background. Okay. A total of 120 trials or 120 stimuli were presented, 50 congruent and 50 incongruent, and these stimuli were presented in a pseudo-random order. Okay, so this is the experimental design for the stimuli presentation. You can see here, this is the stimuli. The stimuli is presented for 1,000 milliseconds, which is basically equal to 1 second. It could be congruent or incongruent, right? And it will be presented in the random order. After the stimuli, a feedback will be given for 500 milliseconds. The feedback is like this: your response is correct or incorrect, and what is that? Or maybe the participant is unable to respond within the stipulated time. This can also happen, right? And then the last is the fixation time, which is 1,000 milliseconds, in which the participant is shown this plus sign, right? So that he gets ready for the next stimuli. So basically, one stimuli, the timing for one stimuli is 1,000 milliseconds plus 500 plus another 1,000. Basically, it is this one, stimuli plus feedback, and this 1,000 is the fixation time. And you can see here from this figure is that the participant is sitting very comfortably in front of the computer. This is the keyboard on which he gives the response, and you see that his EEG, his or her EEG is being recorded when he performs this task. Okay, and since we have all the timings there, so we can relate the EEG with this task performance, and then we can analyze the results. So this is the experimental task, and now I move to the participants. So we have 50 subjects. We hired 50 participants, in 25 in the NFT group and 25 in the control group. And we used the music stimuli also in order to induce relaxation in the participants. And we use the music from Brain FM. If you go and Google it out, you will find that the Brain FM is a company which provides scientifically validated music sounds, like sounds for relaxation, sounds for attention, sounds for focus, and sounds for, I think, sleeping also. So this music is not like the lyrics and the songs that we hear in our movies, like in the maybe the Bollywood movies, that you think, no, not those songs. Those songs are not good for our cognition. These songs are basically the nature sounds. These nature sounds are, we call it music, right? But you know that if these are students, so normally our students nowadays, they sleep during the day and they wake up work all night, right? But I think that's not a very good way of working. So if those of you who wake up early in the morning and let's say they go out for a morning walk, and if by chance you live around a forest or some green area, you will feel very relaxed when you will listen to the birds. Or if you live around a sea or a river, the sound of the water, it is very relaxing sound. If you like the sound of the rain, the sound of the rain is also induces relaxation. So if you go to the Brain FM, you will see that These are all the nature-based sounds which are used to induce different kinds of different kind relaxation or focus. Means actually they induces they enhances the frequency in the human brain, right? So we have used also this music as stimuli. Okay, so what material we have used for EG recording? We have used emotive Epoch headset. It's very commonly used around the globe nowadays. It is a low-cost headset. This is the headset. It has 16 electrode sensors, out of which the 14 are used for sensing, and 2 electrodes are used for as the reference electrodes. These electrodes follow the 1020 international standard, and for EG data analysis, it comes with an Emotive Pro software, which helps to save the raw EG signals. So this is the device that we have used, and now I will move to the because this is a very big experiment, and now we will move to the collection of EG and the behavioral data. So if we look at the collection of EG data, you will see that this is the design of data collection. So our data collection is, you can see that it is divided into 23 visits. So in the first visit, when the participants come, they should give a baseline EG, and then they will fill up some questionnaires like MMSC and PSS, SAM. So these questionnaires they fill up, and then they will do cognitive tasks like Stroop task, and we have another 2 tasks, the attention network task and the Ambedkar task. Attention network task is to check whether the attention is enhanced or not, and Ambedkar is the memory task. But in this work, we will only discuss the Stroop task. And then visit 2 to 11 are the 10 sessions for NFT. Each session is 30 minutes long. After 10 sessions, we will have a further visit in which again the data will be collected, and these events, these Stroop, ANT, and Ambedkar tests will be conducted to see that whether these cognitive functions have enhanced or not. Again, we will go for 10 more sessions of neurofeedback training, and again we will repeat the same process. So the process in the blue is basically looking at collecting the data only, and the gray one are the NFT sessions. And NFT session is also divided into 3 parts, is 15-minute NFT. So different music is used in these 2 sessions, and then after the 30-minute NFT session is finished, then we will collect 3-minute resting state EG, which will help us to analyze the transfer effect of this session. So this is basically the collection of EG and the behavioral data, which is very important for assessing the effectiveness of the neurofeedback. Okay, so once we move this, then we will, okay, so we have now collected the data. All right, so the next step is the cleaning of the data. Means you need to clean everything, right? Any kind of data that you collect, you need to clean it. So now we have 2 types of data: the data that we have collected during the Stroop task, and the data that we have collected during the NFT. Here we will be only talking about the data we collected for the Stroop task. So there is a 4-step procedure. First of all, the Emotive gives the data collected by, because Emotive does not have a very high sampling rate. This EEG headset has a sampling rate of 128 hertz. So basically, the highest signal frequency is around 64 hertz. Then because it's 64 hertz, then we remove the power lines 50 hertz and 60 hertz. So basically, the working frequency in Emotive headset is from 0 to 45 hertz. So that's why we filter the data from between 0.1 to 40 hertz using EEG lamp. So this is the basic filtering, and then we prepare some trials. So we prepare the trials from -300 milliseconds to 1200 milliseconds around the stimulus onset. So basically, one epoch is 1500 milliseconds for the Stroop task. And you know that we have, for one subject, we have 100 trials, right? After that, the data is plotted and each epoch is inspected visually. So it means that you cannot do it automatically. It means it's a time-consuming process. So the researcher has to sit down and he has to visually see that if there is some noise in this data or not. The noise comes from the muscle artifacts because each EEG is captured from the brain or from the head, right? So if you will move your eyes or if you will move your muscles of the face, like you are sitting still, but you are moving your tongue inside your mouth, then again it will create some noise for the EEG, right? So the participants are asked that they should not move any, they should not make any motion, right? Which is not recommended by the researcher. So after that, we remove the artifacts manually. Then what we will do is that we will apply the independent component analysis to eliminate all the independent components. So this can be done automatically, and it can be done using EEG lab. And it's not very difficult if you try to configure it using EEG lab. This process is really straightforward. Okay, so this is the cleaning of the data for the Stroop data. Once we clean data, then we start the pre-processing. So data pre-processing, now you know that the EEG is, now this is another issue, right? EEG is non-stationary in nature, right? So what we have to do is we cannot use the Fourier transform on EEG. It is better that we basically use the time frequency decomposition. That's why for pre-processing, also we use the time frequency decomposition, and we use the Morlet-Wavelet convolution method to have an optimal trade-off between the temporal and the spectral precision, right? So that's why we use the Morlet-Wavelet convolution given by this equation for data pre-processing. It will help us to decompose our signal, right? And then after that, we have to do, in this experiment, we will do two types of measurements. We will do the power measurement and we will do the connectivity analysis. So for power computation, again, for power computation, we need to compute the power over short time windows. So again, we will use the instantaneous power for each trial computed by this equation. So we will use and after that, the next analysis that we are going to do is the connectivity measurement. So we will be using the inter-site phase clustering method for the connectivity measurement. So what is basically inter-site phase clustering is we are trying to find the difference between the phases of the signals on the two electrodes. So how we are going to do this? From the time frequency decomposition, we extract the time series of phase angles for all electrodes. You remember that we have 14 electrodes. So on every electrode, we have a time series. So we will generate a time series of phase angles for every electrode, right? And then we will find the ISPC, the inter-site phase difference. You see the phase difference between two electrodes. Okay. Then cluster the phase difference between the two electrodes, and it will help us to find the connectivity. Connectivity means the connection between two locations inside the brain. So that is the connectivity measurement using inter-site phase clustering. There are many methods to do the connectivity measurement, but this is one of the recommended methods. So once we have done this decomposition, then we will do the how we will going to analyze the data. So we will be, this slide just shows that how we are doing the analysis. We are doing the power analysis and we are doing the connectivity analysis. For power analysis is shorter. We need 3 steps, and connectivity analysis, we need 4 steps. And for both analysis, at the start, we need the time frequency decomposed signal. So the signal is already there. And then what we do in the power analysis is that first we create, we use the power of the signal to prepare this topo map. Okay, this is you can see this topo map. This is the nose, and these are the two ears, right? Ears. So it means that the person is. You can see that where its face is. So, and this is the frontal region. So you see that when you see in this topo map, okay, the red color will show the highest activation, and blue color will show the lowest activation. We will see this in the next slide. So we will prepare this topo map. From this topo map, we will see the region of the highest power, and then we will decompose it we will create a time frequency map. From the time frequency map, we will see the time and the frequency relative to the highest activation, and then we will compile the results. And similarly, when we will do the connectivity analysis, again, what we are going to do, the first step is that we will calculate the all-to-all connectivity, means the connection of every electrode to the other electrode. From all-to-all connectivity, we will threshold this all-to-all connectivity and convert it into the seed connectivity, means the electrode which has the highest density and its connection with the other electrodes. And then again, the same process, the time frequency analysis based on the phases, and then we will do some analysis, two types of analysis. We will do the within the group analysis, and we will do the between the group analysis. So this is basically the layout of how we are going to analyze our data, and which leads us to move to the results. So let's move to the results. So first of all, we will look at the behavior results. So this is the behavior result, because you see that when we do the Stroop task, then we have 100 trials, and we can calculate the response time and the response accuracy. So when we calculated the response time, so if you look at this figure, you will see that the top row is the response time and response accuracy for the NFT group, and the bottom is the control group. So here we see that for the NFT group, the response time has decreased after training. So after NFT training, the response time has decreased, which is a positive effect of the NFT, right? But when we look at the response accuracy, there is no significant change in the response accuracy for the neurofeedback group. So we see that the response time significantly reduced for the NFT group after NFT, and the control group does not show any consistent pattern. So this is one encouraging result, that the response time is significantly reduced. Okay, next we move to the results. The results of the in terms of the power. So we will look at the power of three bands: the theta band, the alpha band, theta band, and the beta band. So this is the theta band power. So first of all, I need to explain this figures, that how we are going to look at this figure. So if we want to look at this figure, let's recall our experiment. We know that our experiment is related to conflict resolution. So when we talk about conflict resolution, we know that conflict related we have a conflict related modulation at the We have a conflict-related modulation at the early stage, and then which is followed by a response-related modulation at the later stage of the stimuli. So, which means that we have divided our experiment into two time stages: an early stage, which is 100 to 500 milliseconds, and the later stage, which is 500 to 800 milliseconds. And you see that this is theta band, which is 4 to 8 hertz. And then we have, now we have studied 4 cases in order to look at the congruency effect. You see that we have two types of stimuli, the congruent and the incongruent stimuli. So when you look at IC means the incongruent stimuli is followed by a congruent stimuli, right? when we look at CI, it means the congruent is followed by incongruent. So this is known as congruency alternation, because in two stimuli, the congruency is altering. Okay. And then we have congruency repetition. Repetition means when you have the two stimuli, they are same, the congruent and congruent, and incongruent and incongruent. So it means that how we are analyzing our data is that we have divided it into two time windows and two types of congruency effects: congruency alternation, the different effect, and the congruency repetition. So we have 4 topomaps. Then we will combine the congruency alternation topomaps, these two topomaps into one topomap, these two into one, right? And these here and this one is here. So this is the combination, right? So after plotting these topomaps, we combine into these topomaps. Now we have 4 topomaps. If you look at this topomap, now what we can see from this topomap? Let's again look at this topomap and look at this bar. So this bar tells us the power of the signal. So if you have dark blue color, the least power, and the very deep red is the highest power. So from this topomap, we see that most of the highest power is present in the frontal region. This is the location of the A4 electrode, and we also see that another very high power is present in the occipital region. Occipital region is the region which is responsible for viewing the. Basically, it is related to the sense of the human sense of sight, right? So this is A4 and this is O2, right? And if you look at this is also same kind of activation. If you look at it here we see that this activation is basically in temporal region, and this activation is in the frontal AF3 region. So the right side of the brain is normally given the even numbers, and the left side of the brain is given the odd numbers. Okay, so once we make these topomaps and we see that where is the high activation, then we select the electrodes. We have already known the electrodes, and what we do over here is that we again plot the time frequency plots, right? And from the time frequency plots, you see that based on the visual observation, we see that which region is the highest activation region, right? So highest power is present in this region, and here the highest power is present in this region. So then we will do the analysis. We will look at this region, and then we will do the analysis. We will analyze the results. So here we will you see that this is the result of our analysis. This is the NFT group, and this is the control group. And for the theta power, we see that if you see that the red line represents the NFT group after post NFT, after all NFT sessions. So here we see that the theta power has increased after NFT. Here also we see that the theta power has increased after NFT. But for the control group, it is the reverse, right? The theta power didn't increase after NFT. But although in the NFT group, the theta power has increased after NFT, but the result is not significant, right? The result is not significant statistically, although the power has increased. So this is the result of the theta band power. And now if we move to the alpha band power, because we are following the alpha protocol. So the procedure is exactly same. Here you see that. So I will finally directly go to the results. So you see that when we talk about the alpha band power, again, we see that post NFT, in the NFT group, the alpha band power has increased. Okay, from both figures, we can see it is increased. And then when we look at the control group shows a varied pattern, right? Sometimes it's decreased and sometimes it's increased. But when we do the statistical analysis, we see that the increase in the alpha power in the NFT group is is not significant. Okay, all the power has increased, but the increase in is not significant. Okay, and control group showed no pattern. After that, we looked at the beta power, because beta power is related to also attention. But when we looked at beta power, even in the NFT group, the beta power showed a varied pattern, means we couldn't identify the pattern neither for the NFT group and nor for the control group. And also this change is not significant. So this is the effect of NFT on the three different powers. And now we will look at the results which are related to the theta band connectivity. So again, let me explain this figure first. You remember that we have an early time window, which is because of the conflict related modulation, and we have a late time window which is related to the response related modulation. So first of all, what we did is that we plotted the similar to the power analysis. We plotted the we have the alternation case and the repetition case, right? And then what we have done, we have plotted the all-to-all synchronization. What is meant by all-to-all synchronization? It means that we have calculated the strength of the network between each electrode, right? So if you look at these balls which are plotted here, the color and the size of the circles show the connectivity degree at each electrode. So the color and the size. So if the size is big, then you see that the maximum connectivity is 5 and the minimum connectivity is 0. So you see that if it is lots of connectivity here, right? In the frontal region, you see lots of connectivity for the alternation case in the late time window. But for the repetition case, you see that the connectivity is less than the frontal, than the alternation case. And basically, maybe why? Because for alternation case, you need to focus more. So you need more connectivity, right? So this is the all-to-all synchronization. But in order to get results, we need to find the seed synchronization. So from here, what we do is we need to move to this figure. And in order to move to this figure, what we do is that we will binarize the connectivity matrix using a threshold, and then we compute the node degree for each electrode. So you see that here, these values are different. We have computed the node degree for each electrode. There are 14 electrodes, so the node degree can be 14. And this is the seed electrode. Seed electrode is the hub electrode, which has the highest number of connections. Anyway, for this particular case, we have chosen the node degree in such a way that it does not exceed. It does not exceed. It is not more than 5, right? So based on the node degree, we choose the seed or the hub electrode, the black one, which has the maximum node degree, and with the hub electrode, the seeded connectivity is computed. So you see that in this figure, you see that every electrode is connected to the other electrode, or not every, but many. But here you see that we have a seed electrode. So this we have wired the connectivity of this seed electrode with the rest of the electrodes. That is a synchronization of the hub to all electrodes. So after we calculate the seed electrode, then the electrodes with maximum node strength with the hub electrodes are is this seed electrode, and the one electrode which has the maximum strength will be used for the analysis, not all electrodes. Okay, so again we will plot the time frequency plots with maximum synchronization with the hub electrodes, and then visually we will choose the maximum synchronization regions. And once we have the maximum synchronization regions, then we will make the results, analyze the results. So this is the results for the NFT group, and this is the result for the control group. And now you see that the theta band connectivity has improved a lot in the NFT group post NFT, and we see that this is also increase in the connectivity is significant for the control group, but it is not for the NFT group, but it is not significant for the control group. Next, we move to the alpha band connectivity. So again, the same procedure that we have followed, and then we have prepared the all-to-all synchronization. Then we have calculated the seed node synchronization, and then we generate the results. So again, for the alpha band connectivity, again we see that the post NFT results are better. Connectivity is high, and it is significant. Okay, but for the control group, there's the connectivity is decreased, right? So, and it does not show any specific result. And finally, we calculate the beta band connectivity. The same procedure we have followed. So we move to see the results, and then we look at the beta band connectivity. We see that beta band connectivity is not increased post NFT, right? Actually, it has decreased post NFT, and whereas in the control group, it looks like it is increased, but this increase is not significant, nor the change in the connectivity of the beta band in the NFT group is significant, right? So these are the results of the connectivity for the 3 bands, and we have seen the results for the power for the 3 bands, and now we move to our summary of these results. So we see that the results show an increase in the theta and the alpha band powers and connectivity. Okay, we have seen that the theta and the alpha band powers have increased, well as the connectivity also increased. We have seen that there is no training specific modulation observed in the beta band functional connectivity. Okay, so we can say that a significant improvement in the theta and the alpha band connectivity is observed in the NFT group, which is not present in the control group. Okay, so this is basically the summary, and which brings us to the conclusion. So what we have done here in the current study is that we have used a non-invasive neurofeedback training on healthy participants and investigated the effect towards response conflict resolution. With the help of the EEG recordings and employing the time frequency analysis method, we investigated the evidence of NFT-related differences in healthy participants. The participants showed improvement in the processing speed, means the behavior results was good of the response conflict task, which is indicated by the faster response time in the NFT group. And we also found that the alpha power in the response conflict task indicates training related differences at the frontal sides. Also, theta power shows NFT related changes at the anterofrontal, occipital, and temporal sides. So basically the conclusion is that the behavior means the response time has improved, and the alpha and the theta powers have been as well as the alpha and the theta connectivity shows some positive results. So which means that we can say that this alpha protocol based NFT training has some positive effect. The theta and the alpha band phase synchronization between the frontal and the parietal areas of the brain enhanced in the NFT group also. So this impact is not seen in the control group. Hence, our findings suggest that the neurofeedback training is a positive cognition enhancement procedure for healthy adults. For this specific case, we are talking about the response conflict. Okay, so this concludes my talk, and then for future consideration, we can say that the participants of the current study were recruited from the university populations. For generalizing this study, one of the future possible extensions is that we can extend this study to the healthy people, but from the general public, because you know that the students who who come to the university are very privileged students, right? Not the general public. Not everyone is privileged. So it may be possible that those general public, after they are exposed to this kind of training, maybe they show more improvement as compared to the already trained students who study in the university, right? And then we can also extend this study to neurological, the people who are facing neurological problems. Okay, so this is all from my talk, and these are the list of references. This talk is basically based on reference 12, which is a open access paper. You can download it from IEEE Access. These are my team members, my collaborators, and I would like to acknowledge the UTAR RF Research Fund, as well as the FRGS Fund for this work. Something is wrong. Everything is okay, right? Okay. Okay, so this is the end of my talk. I thank you everyone for listening to me, and if you have any questions, you can ask now.
Thank you, Ma. Participants, any questions, any queries? Can we ask
There's a
Madam?
question from the chat box.
Yes.
It states that does the theory hold feasibility in all living animals, similar to humans?
Hi, hello, please can you repeat?
Yeah, yes, ma'am. In the chat box, it is there like, does the theory hold feasibility in all living animals, similar to humans?
I have not. I am not aware of studies on the animals. I'm very sorry. So the studies that I have gone through are all on the human beings. But I know that there are some studies on animals also. But since I don't work with the animals, so I'm sorry, I'm unable to answer this question.
Yes, madam.
Participants, any queries?
Yeah, my name is Borchala from Ethiopia.
Yes, sir.
Yeah, thank you for valuable information, Mr. Maniara. Yes, madam.
Yeah.
Am I audible?
Yes, I can hear you.
Yeah, it is an interesting topic. Also, I am graduated in electrical and computer engineering. My research area is on this heart rate measurement from fingertip. From that, I take this. Can you mention for me EEG, electroencephalography, and ECGS? Between them, can you mention it?
Actually, EEG is the signal from the brain, and ECG is the signal from the heart, right? So in some studies we use multimodality signal, but they are two different modalities, right? Although they are both physiological signals.
Yeah, look. How another question? How do you are seeing this from the current era and the current topic? How do you see this? You are what you get from that?
Okay, if I understand your question well, I think this is a very growing research area. So you can always combine. Basically, you can combine the ECG and EEG analysis for different types of for analyzing many things. Like emotion recognition is normally used. For emotion recognition, we can use the ECG as well as the EEG signal. Both modalities can be used.
Yeah. So thank you, ma'am. Then for the future, can you put your mail in the chat box? Because I have a query for the future.
Yeah, sure. Okay.
Yeah, put on the chat box. Thank you.
Ma'am, there's one more question. Is there any difference in the pattern of male and female?
Okay, so there are some studies which shows that there is So, because this is a very interesting question, right? People think that the male brain is different from the female brain, right? But sometimes when we do the analysis, we have not find, because this human brain thing is a very complex, human brain is a very complex structure. So for and when you talk about neurofeedback, it is basically related to the training. You need to modulate your brain, right? So we have observed, irrespective of the male and females, we have observed that some people are better in modulating their brain activities, right? There's, in simple words, some people are better in neurofeedback training and some people are not very good in the neurofeedback training. But we have not seen any differences between the male and the females, that are males good in modulating their brain activities or females better in modulating their brain activities. We have not seen this difference. Means you can see some studies in which they say that, okay, this group of subjects are better, but this study cannot be is not valid for the whole population, because in some studies, in my another work, we used video games to improve the we use the neurofeedback games for stimulus. We didn't use the music. We used the car racing game, and we saw that in that study, the females there was one female which has a very good results. So we cannot generalize yet, right? But although using the brain signals, you can differentiate between the genders. It is possible. Like if you classify the genders based on their EEG signals, it is possible. You know that nowadays these deep learning methods have been very useful in classification. And yes, you can. It means that the EEG of males and females can be different. That's why it can be classified, right? But specific effect on neurofeedback training, we don't know.
Okay, ma'am, there's one more last question. What are the hardware requirements for EEG signals?
Okay, so specifically hardware requirements, you need, there are many EEG devices. There are low-cost EEG devices, then there are research-based EEG devices, and then there are clinical EEG devices. So you know that previously this EEG, this field of neurosciences was not open to the general public, right? General public, not in the sense of public like general universities. But now, because initially the devices which were used for in the clinics, means in the hospitals, basically you remember that you may have heard about the MRI, right? The MRI is often used for the EEG connectivity analysis and this cognition analysis, but MRI is very expensive. Everyone cannot use it. But then after that, there are more EEG devices which were non-invasive, just like this headset that I have shown you, but they are known as a research-based, research-grade EEG devices. These devices have a very high resolution. They have very high number of electrodes. Like they have, they may have as many as 256 electrodes or 128 electrodes. It means that they can cover your scalp very nicely, which means that you will get lots of high amount of data. And then these devices have very high sampling rate. Sampling rate means some of these devices have a sampling rate of 500 samples per second. It means that the signal frequency will be at least 250 samples per second. But in the case of emotive has a very, very low sampling rate, right? It has a sampling rate of 128 samples per second. So the signal frequency is only 64 hertz. So this is a limitation for emotive and many other low grade devices. But when we I believe you understand why this sampling frequency is important, because when we talk about the cognitive tasks are the tasks which have a very high time resolution. Means, have you ever thought, we never think like this, that how you are moving your hand. I moved my hand right now. It takes milliseconds to move my hand, right? So it means that if my sampling frequency is very high, then only I can track this task, right? That a signal is generated in my head and I move my hand in milliseconds. So that's why the high sampling frequency is very important. And the low cost commercial devices, which are basically made for gaming, just like Emotive, and there are so many other devices, right? So initially they were made for gaming, but later many researchers, when they analyze them, they say that, okay, they can also be used for the research purposes. So that's why now this field of research is open to many people around the world because of the low cost devices. Otherwise, the high cost devices, most of the universities around the world, they cannot afford to buy those high cost devices, which are very expensive. So yes, there's a difference between these devices, but you just need to do a good review, and you can see that what kind of devices you can use and you can buy, and depending on your application. So nowadays there are many devices, and more devices are coming in the market. So I hope I have answered your question.
Yes, ma'am. Thank you. I think any queries?
So I don't see. I have written my yes, I have written my email in the chat box.
Yes, ma'am. Have you written?
And there's no more question, right?
Yes, ma'am. Yeah. Let me just.
This is only one question: hardware requirement.
Yes, ma'am.
Can we get PPT? Hello.
Hello. Yes. Because I think the video is being recorded, right?
Yes, ma'am. We will share the video.
Okay.
The WhatsApp group, if possible.
Sure. We'll just do it.
Yeah.
We shall just do it. Okay. Okay, ma'am. Good afternoon to one and all. Myself, Dr. Shilpa Casey, Assistant Professor, Department of Electronics and Communication Engineering. Feel glad and highly privileged to present the vote of thanks on this occasion. First and foremost, I would like to extend my heartfelt thanks to our guest and the speaker, Dr. Umaira Nisar, Associate Professor, Head of the Program, Master of Engineering, Electronic Systems, Faculty of Engineering and Green Technology, University Tunku Abdul Rahman, Malaysia, for sharing her knowledge with all of us and throwing light up on effects of neurofeedback training on response conflict. I take this privilege to thank our management members on behalf of our department for providing us the opportunity to conduct the webinar. I wholeheartedly thank our beloved principal, Dr. G. Rajendra, who has been very supportive throughout and guided us with his suggestions in organizing this program. I thank the director of our mentee institution, Dr. Ramapati Mishra, to be a part of this event. I take this opportunity to thank all the deans and heads of the respective departments who have accepted our invitations and are present here to encourage us and our participants. I thank Professor Vasudev Murthy, IST coordinator, who has been the driving force in the organizing this department. I'm very grateful to thank Professor S. Ramesh, Professor and Head, Department of Electronics and Communication Engineering, for being very keen and enthusiastic for organizing this webinar. I extend my warm regards to all the faculty members from various departments who are present here and witnessed this inaugural session and keynote address. Next, I would like to thank my fellow coordinators, Sangeeta N, Shweta N, Siddesha K, who has been working from the beginning for the smooth conduction of this event. I would also like to thank Mr. Jarit Gupta, Assistant Professor, Department of Computer Science Engineering, KIET Group of Institution, Ghaziabad, Uttar Pradesh, for providing us the technical support. Last but definitely not the least, I thank all the participants who have registered to this training program with a great enthusiasm to learn about this topic. On behalf of the coordinators, I would like to tell that we are equally excited as you're all, and I'm sure that we will all try our best to reach your expectation. Thank you, one and all. Have a nice day. Stay home, stay safe. Thank you. And the feedback link is posted in the chat box as well as in the respective mail IDs. Kindly to fill it to avail the certificate. Thank you, one and all.
Thanks, Shilpa madam. Thank you very much.